In 2005, epidemiologist John Ioannidis published a paper with the provocative title, "Why Most Published Research Findings Are False." His argument was mathematical. Given typical study sizes, typical effect sizes, and the p-value threshold of 0.05, the expected proportion of false positives among significant findings is much higher than most researchers realize. Statistics can be a tool for discovering truth or a factory for producing impressive-sounding noise. Statistical hypothesis testing begins by formulating two competing hypotheses. The null hypothesis is the default assumption that there is no effect, no difference, and no relationship in the population. The alternative hypothesis proposes that a real effect exists. The logic of null hypothesis testing is indirect. Rather than proving the alternative hypothesis true, we ask how unlikely our data would be if the null hypothesis were true. If the data are very unlikely under the null, we reject the null, not because we have proven the alternative, but because the null is implausible given what we observed. The p-value is the probability of obtaining results as extreme as or more extreme than those observed, assuming the null hypothesis is true. A p-value of 0.03 means that if the null hypothesis were true, results like these would occur only 3% of the time by chance. The conventional threshold for rejecting the null hypothesis is p less than 0.05, meaning you accept a 5% chance of being wrong when you declare a result significant. The p-value is not the probability that the null is true, nor the probability that the alternative is true, a crucial distinction that is frequently misunderstood. A statistically significant result means only that the observed data are sufficiently unlikely under the null hypothesis to justify rejecting it, typically at the p less than 0.05 level. Statistical significance is not the same as practical or clinical significance. A study with 10,000 participants can find a statistically significant difference that is so tiny as to be completely meaningless in practice, because larger samples provide more statistical power, making it easier to detect even trivially small effects. Always evaluate effect size alongside p-values. Every statistical test carries two types of potential error. A type 1 error, also called a false positive, occurs when you reject a true null hypothesis, concluding there is an effect when there is none. The probability of a type 1 error equals the significance threshold alpha, typically set at 0.05. A type 2 error, or false negative, occurs when you fail to reject a false null hypothesis, missing a real effect. The probability of a type 2 error is called beta. The complement of beta is statistical power, the probability of detecting a real effect when it exists. Internal validity is the degree to which a study can support a causal conclusion, the extent to which changes in the dependent variable can be confidently attributed to manipulation of the independent variable rather than to confounding factors. A study has high internal validity when the only systematic difference between experimental and control conditions is the independent variable. Random assignment of participants to conditions is the primary tool for achieving internal validity, as it distributes potential confounders across conditions by chance. External validity asks whether the findings of a study generalize beyond the specific participants, setting, and context of the study. A study with very high internal validity, tightly controlled laboratory conditions, may have low external validity because it is so artificial that its findings do not apply to real-world situations. Research conducted with weird populations, Western, educated, industrialized, rich, democratic, may not generalize to the majority of the world's population. Ecological validity is a related concept referring specifically to how well the experimental situation resembles real-world conditions. A single study, no matter how well designed, cannot establish a finding as reliable knowledge. Replication, the independent repetition of a study by different researchers to see if the same result emerges, is the gold standard for scientific credibility. The replication crisis in psychology, formalized by the Open Science Collaboration in 2015, found that only about 36% of 100 significant psychology findings successfully replicated. Factors contributing to poor replicability include small sample sizes, publication bias favoring significant results, p-hacking, and undisclosed flexibility in data analysis. Experimenter bias occurs when the researcher's expectations inadvertently influence the results of the study. Robert Rosenthal demonstrated this with his famous study of rat learning. Experimenters told their rats were specially bred for superior maze running reported significantly better maze performance, even though the rats were randomly assigned. Rosenthal and Jacobson then showed the same effect in classrooms. Teachers told certain students were on the verge of intellectual blooming showed measurable gains, compared to equally able students not labeled as promising. The double-blind procedure, in which neither participants nor experimenters know condition assignments, controls for this bias. Demand characteristics are cues in the research environment that suggest to participants what behavior the researcher expects or desires. Participants, wanting to be helpful or to present themselves positively, adjust their behavior accordingly, not because of the independent variable, but because of their interpretation of the experimental situation. Demand characteristics threaten internal validity because the behavioral change is caused by social dynamics rather than the four. Single-blind procedures that keep participants unaware of condition assignments are the primary control. Publication bias is the systematic tendency for journals to publish studies with significant positive results while rejecting or ignoring studies with null results. This creates a distorted scientific literature in which the published record substantially overestimates effect sizes and the prevalence of significant findings. If 20 researchers test the same false hypothesis, one will find a significant result by chance at alpha equals 0.05. And that one result is the one that gets published, while the 19 failures go into the file drawer. Pre-registration of studies, publicly documenting hypotheses and methods before data collection, is the primary reform designed to counteract publication bias. Statistical thinking is probabilistic thinking. It means holding conclusions with appropriate confidence rather than false certainty. A significant p-value is not proof of truth. A replication failure is not proof of fraud. The scientific process is self-correcting, but only when researchers understand what their statistics actually mean and report their findings honestly.